I would like to share about our speaker that's coming up on stage. I was at his small group session yesterday, and he shared that he shared two takeaways for many of the students who were there. There were over 20 of us, and he said, like, first of all, if you're not happy, you must complain. And secondly, you must take advantage of opportunities. If you're lucky, don't feel bad about it. Luck has come your way, take advantage of that. And the person who shared that with me and the 20-odd students that were in that small group session was none other than Professor Sir Richard Roberts. Please put your hands together and give him a warm welcome. He will be talking about the path to the Nobel Prize, and the Q&A session will be moderated by Professor Polly Chen from the National University of Singapore. Sir Richard.
Okay. So, thank you very much. Always a pleasure to come to Singapore, but even better when one gets to meet students, because I'm a very big fan of students. What I'm going to do today is to talk about my life's journey, if you like, and how it eventually led to a Nobel Prize. And along the way, I want to tell you about a few things I learned that I think students often find useful and helpful. And this last point that was made about luck, I will illustrate that in many different ways during the course of the talk. And I'm going to start by telling you that I was born in Derby towards the end of the Second World War. And the street that I lived on was on a direct path from Germany to the Rolls-Royce factory, where they were making all of the engines to fly over and bomb Germany. And the bombing at that time was not very accurate, and so quite a lot fell on the street where I lived. But the side of the street that my house was on didn't get bombed, and the other side almost got completely destroyed. That's luck. This is the story of my life, is luck, and we'll talk more about it later on. When I was 4 years old, my father moved from Derby here down to Bath, which is in the southern part of England, very beautiful city. If you go to the UK, if you get the chance, go and visit. Beautiful, beautiful city. You just need to spend a day there. You can walk around, see everything. It's all walkable. Very interesting place. When I was in Bath, I went as a junior to a junior school called St. Stephen's School. Very small area. There were 4 classes, each with about 30 people in. And something very important happened to me there. And the thing that happened was I used to leave in the evening, at the end of the afternoon, and the headmaster would come over and would give me a puzzle to do. And they were almost all mathematical puzzles. And I loved them. I thought these were just great. And I fell in love with mathematics as a result of that. And I always thought that, he did this to everybody. But he didn't. He was just doing it to me, it turned out. And he must already have seen something that other people didn't see in me at that time. But he did me a very big favor. His name was Mr. Brokes. And I'm very happy to say that he was obviously a headmaster. He was much older than I was. But he just died about a month after I won the Nobel Prize, and someone had gone and told him that. And I'd shared this story with a journalist. So he realized, I hope anyway, how important he was. Now, after the junior school, I read a book. It was a book, How I Became a Detective. And I thought this sounded fantastic. A regular police detective. I thought this sounded fantastic. And I was absolutely convinced. Previously I'd thought I was going to be a mathematician, but now I was going to be a detective. I was absolutely swept away by this. And then my father came along and bought me a chemistry set. And it was in the times when chemistry sets were interesting. Now the government have taken all the fun out of chemistry. But it was possible to make explosives. You could make fireworks. I loved it. I understand why terrorists like explosives, believe me. So that was really good. And he built a little chemistry lab. It was just really a table with a Formica top. And he helped get chemicals from the local pharmacist and got various bits of equipment. The pictures here, this is the Roman bath. Bath is built on hot springs. and the Romans had found it and built these beautiful baths, and then the Georgians came along and built these really beautiful crescent. This is called the Royal Crescent, but there's so much architecture like this, all using local limestone. Now, when I was 11, you take something called the 11 plus exam, and it was the most interesting experience because we just showed up at school one day and we were sat at a desk and we were given a few pieces of paper and said, can you just answer all of these things? Which we did. We had no idea we were taking an exam. And it's absolutely the best way to examine people. And I learned later, many, many years later, when they made me an open citizen of Bath, that I got the highest score in mathematics on that time. So that was good. But we had no idea. We didn't know we were taking an exam. And so that's really good. I like that very much. When I went to this high school, grammar school, we had a great math teacher, and so I loved that. I also learned to play the violin, and I've had a lot of enjoyment about that over the time. I'm not a good violinist. I got to practice for hours every day in order to make it even sound good to me, but I got a lot of enjoyment out of that. And we live close to the Mendips, which is a limestone escarpment, and I got interested in caving, used to go underground all the time. And I also love jazz, because Bath is rather quite famous for jazz. But an interesting thing happened. When I was 17, we had taken all of the courses necessary to take what they call the advanced level examination to see whether you're going to university or not. And I failed physics. And that was a little disaster. I ended up having to spend another year. There were two reasons I failed physics. One was the teacher was hopeless. He was really useless. And so he couldn't really, get you engaged in it. And then the other thing was, when I was 18, I stopped going because it turned out the first year in the sixth form, they did all the new stuff, and at least it was halfway fun. But the second year, they just repeated it all, and so I stopped going. I used to go and sign in first thing in the morning, and then I'd go down to the local YMCA where they had a billiards table, and I would play billiards and snooker all day, and then I would go and sign in at the class at the end of the afternoon, so they thought I'd been there all day. Well, there was one of the teachers who was a real pain, and he didn't like me, and so he found out what I was doing, and it looked as though I was going to get expelled. And the thing I did best at the time was play snooker. I was the West of England junior snooker champion, and I went to audition to be a professional snooker player because the world champion at the time, Joe Davis, was looking for a sparring partner. He went around doing exhibition matches and so on. And so he was looking for someone, and I was pretty good. I went, but I never found out whether I was going to be taken because the school relented and said they were not going to throw me out. So I always thought I missed the chance to be really famous. I could have been the world snooker champion. But I also learned an incredibly important lesson. I was playing snooker one day in a tournament, and I was trying to pot a ball, and I missed the ball that I was trying to pot, but I got another one instead. And I played the next shot, and then I just fluffed it because I just wasn't concentrating hard enough. And afterwards, an old gentleman, about 80 years old, came up to me and he said, "You could be a really good snooker player," he said, "but you have to learn when you get a lucky break, you've got to make the most of it. You've got to concentrate twice as hard on whatever you do next to make sure that you took advantage of the luck." And everybody has luck. If you look back on your lives, you'll see where luck led you this way or that way or the other. And I learned a valuable lesson then, and from then on, whenever something lucky happens, I always see how I can capitalize on it and make the most of it. When it came time to go to university, because I had failed physics the first time and had to take it again, I wasn't eligible for Oxford or Cambridge, and I wanted to do chemistry, and they actually were not the best places to go do chemistry anyway. There were two good universities, Southampton and Sheffield. I applied to both. Southampton turned me down. Sheffield accepted me, so I ended up going to Sheffield. I had a very good time. I did chemistry as the major there. I also discovered that if you teach physics properly, it was actually enjoyable. I could do it. I had no trouble whatsoever. And I decided I was going to pursue a career in research because it allows you to be a detective and you're solving puzzles all the time. And, you know, it's just great. Everything I love to do, all of a sudden I was able to do it by doing research. I got my BSc in 1965, and I had an interesting experience then. They took something called the pre-final exam after 2 years as an undergraduate, and I got a 2-1. You know, a first class is the best, and 2-1. And my tutor came along and he said, you know, if you just worked a little harder, you could get a first. And I thought that sounded pretty good, and especially as I'd done absolutely no work whatsoever to get this 2-1 in the first place. And so the third year at university, I worked my ass off. I studied everything you could imagine, and then I ended up getting a 2-1. And I decided that this was not the way to pass exams. It wasn't worth it, you know. I was perfectly happy with a 2-1. And I wanted to do research. I stayed and worked with Professor Ollis. He was head of organic chemistry. And he was great. When he set exams, they were puzzles. And so, you know, it just played into my strength. I love solving puzzles. I play puzzles all the time. I love word games. I love games. It's great. And so when I went to start being a PhD student, another piece of luck came my way. This gentleman in the middle, Kazu Kurosawa, was a Japanese postdoc in the lab where I was working. And he is probably the best mentor that I've ever had in my entire life. He was fantastic. He would tell you how you should do the experiment, why you were doing it, why you do it this way rather than that way. Wonderful at explaining results. If things work, then he would explain why they worked. If they didn't work, then he would explain what you'd done wrong. And he was fantastic. And he not only taught me a lot of chemistry, he also taught me how to play Go, and that's a game here, which is actually a game that's much more difficult than chess, much more complicated, but it's a wonderful game. If you've never tried it, you should try it. And he and I used to play Go by mail after he left and went back to Japan. And we did that probably for 3 or 4 years until his English got so bad, it was clear it was difficult for him to keep writing the letters, you know, that he felt he had to write to accompany the moves. Anyway, so that was good. Now, because of Kazu, I had all the results I needed for my thesis in 1 year. At the end of the first year, I could easily have written my thesis, but you know, this was England, and the Brits don't let you graduate in 1 year. You've got to do 3 years. So the second year, I played around doing things that I thought were good. Didn't really go anywhere, and a lot of failure during that time, which is okay. Failure is good. And then I went to the library and started reading. And I started reading because I was thinking about what I might do next. And I went to the library and discovered that there was a journal of molecular biology, and there was some other stuff about it. And I read this book by John Kendrew. John Kendrew got the Nobel Prize for determining the first protein structure. And this book was all about how molecular biology got started. And it was so good. By the time I finished the book, I knew this was what I wanted to do with my life. And it's interesting, I wrote that in my Nobel autobiography, and John contacted me, he sent me a copy of the book, and he said this was the nicest thing that anybody had ever said about his book, because apparently it got terrible reviews. But for me, it was all I needed. It was great. So I applied to 6 labs to switch from chemistry into molecular biology, and I picked labs that were not sort of mainstream molecular biology, but were sort of chemistry that was leading into molecular biology. Of the 6 labs, only 1 accepted me, and that was Jack Strominger. And at the time, he was at Wisconsin, and I thought I was going to have to go to Wisconsin for my postdoc. And about 2 months before I was scheduled to leave, he wrote to me and he said, "Can you delay coming for a month because I'm just moving to Harvard?" And so again, lucky break, by chance, I ended up at Harvard. I'd applied actually to somebody else at Harvard for a lab, and they turned me down, but he accepted me, so that was good. And the problem that I worked on was transfer RNA and learning how to sequence transfer RNA. And the key thing there was, here was transfer RNA usually takes an amino acid and puts it into a protein on the ribosome. Okay? This particular tRNA didn't work in protein synthesis. It only worked to put glycine into the bacterial cell wall of Staphylococcus epidermidis. And the question was, why? What is it? What is the sequence? And so I picked up this project from a postdoc Tom Stewart, who'd already started. He was an Australian. He left after about 6 months, went back to Australia, and I realized that the method he was using was a sort of old-fashioned method. It was one that Robert Holly, had won the Nobel Prize for developing. But I was reading about this method that Fred Sanger had come up with. This is Fred Sanger here under the red arrow. Fred, an amazing person. He's someone I would love to be like. He's the most modest man I've ever come across. He won a Nobel Prize for developing protein sequencing, then won a second one for DNA sequencing. He could have had a third for RNA sequencing because he developed that too. I went to his lab, spent 6 weeks there, came back, set up a sequencing lab at Harvard, and turned out, by chance, by luck, I was the first person to be using Fred Sanger's methods in the Boston area, and everyone came to me to learn how to do it. I made a lot of really good friends in those days. One of those friends was an assistant professor at the time at Harvard called Mark Potashny, and he came up to me one day in a seminar and he said, you're looking for a job. Jim Watson would like to talk to you. He's going to come and find you because he needs someone to go to Cold Spring Harbor and sequence SV40. That sounded interesting. I knew, obviously, who Jim Watson was, Watson and Crick and the double helix and all that. But I also applied for a couple of other jobs too. One in Edinburgh that I was told I was perfect for. I sent in an application, never heard back. I went and interviewed at Harvard to be in the biology department, and they offered me a job, and then Jim Watson. So Jim Watson never came and found me, and so Mark came up one day and he said, Jim doesn't actually know who you are, so maybe you can go and introduce yourself, which I did. Go into his office. I write here it was a 10-minute interview or whatever it was. It may not have been that long. I went in. First words were, I want you to come to Cold Spring Harbor and sequence SV40 DNA. And then we chatted on about other stuff. He said, oh, by the way, you probably should come down to Cold Spring Harbor and see what it looks like, which I said, okay. And he said, but it's not an interview, he said. It's not an interview. You got the job. It's yours. And then he kicked me out of the office. So that was okay. I did go down to Cold Spring Harbor and check. It was a rainy day. I had one decent set of clothes, and he took me walking around the grounds. I was up to mud in my knees by the time I got over, and then I had to give a little seminar, so that didn't go quite so well. Anyway, when I got to Cold Spring Harbor, what he had in mind was to sequence SV40 by copying it into RNA and then sequencing the RNA, because this was how people were doing it those days. But I discovered there were 2 other labs doing exactly the same thing. There was Sherman Weissman at Yale was trying to do it, and Walter Fiers in Ghent, Belgium was doing it, and they were both using these methods. And, I don't like to sort of just set myself up for a competitive situation. And I thought that I was going to find a new way to sequence DNA. And the reason I thought I could do that was because just before I left Harvard, I went and found out about restriction enzymes. This man, Dan Nathans, in the top left corner, won the Nobel Prize for the restriction enzyme work with Werner Aber and Hamilton Smith. And he gave a talk explaining that these restriction enzymes, or just the one actually that he was using, endonuclease R from Haemophilus influenza, could cut SV40 into small pieces. And I thought, this might be the way to start tackling DNA sequencing. And so when I got to Cold Spring Harbor, I told Jim I wasn't going to sequence SV40 the way he wanted it. I was going to study restriction enzymes and see if we could develop a sequencing method. That got him completely pissed off. And I think I spent 20 years at Cold Spring Harbor, and I don't think he had a really good word to say for me about the whole time I was there. We used to fight all the time. But I had a good friend who helped me. So that was working out okay. And so then we started a project to look at the promoter sequences in adenovirus. Adenovirus is a eukaryotic virus. We started trying to look at the sequences of the early RNAs coming in from the ends, couldn't get enough RNA, and so then we started working on all the late RNAs. And the idea was if we could find the 5' T1 oligonucleotide, T1 ribonuclease, T1 cuts after G, if we could find these 5' termini, then we would know that sequence just upstream was going to be the promoter. And so we did that. I had a postdoc, Richard Gelinas, who came, did the experiment, and made late adeno messenger RNA and discovered there were 2 spots when you did a 2D gel analysis. And if you look late in infection, when there was only late RNA present, there was just 1 spot. All of these RNAs from the previous slide, they all had the same 5' end. We published this, I hope, yeah. So 1 predominant 5' undeconucleotide. But the question was, why did they all have the same ends? And so we came up with loads of experiments. We spent about a year and a half trying to figure out what was going on. Because basically this experiment to catch these 5, it was a super experiment. Everybody agreed it was. I had thought of it, so of course I knew that it was a great experiment to do, but it didn't give the right result. And so we spent a long time doing it. And one thing that came up was that maybe there is a primer RNA that is starting transcription in adenovirus, and we knew there was an RNA made by adenovirus. And so we checked that, and the method that we chose to do it, the experiment, is you take DNA, you take messenger RNA, you hybridize the 2, and at the 3' end, all eukaryotic messenger RNAs have poly A, and at the 5' end, there's a 5' triphosphate. And if this was not coded right next to the gene, then we should be able to find it if we could find the right piece of DNA that would code for it. And the right piece of DNA we thought would be this VA RNA, so we made this. And I came up with this experiment on a Saturday. It's an EM experiment. You have to use electron microscopy to see what's going on. And we were not, neither Richard nor I knew how to do electron microscopy, but we had 2 friends who worked in the lab on the floor below us, Tom Broker and Louise Chow. And so we went to them and we said, here's the experiment we want to do. Could you do it for us? And they said, well, it's never quite been done, but yeah, we'll give it a try. It seems as though it should work. So Richard made the reagents we needed on the Sunday and the Monday, and we went down and gave them to Louise Chow, who did the actual experiment. And the very first thing that she saw in the electron microscope looked exactly like I had drawn it on the board. Just one small difference. Instead of hybridizing at just one place, it was hybridizing at 2 places. And it was hybridizing at 2 places because of RNA splicing. And so that led to the discovery of RNA splicing. In the afternoon, everybody was talking about Nobel Prizes because it was clear there was something going on here that was very different from the way in which bacteria make messenger RNA. So that was very exciting. We published this paper in Cell, and it has the word amazing here in the title. Had a lot of trouble with my co-authors. They didn't want to put it in, but eventually they agreed. We sent it off. The reviewers, 3 reviewers, all wrote back and said, you can't use amazing, this is not scientific, it's wrong. And so I called the editor, and the editor said the same. I called the editor and started talking to him about some of the things that the other reviewers commented on, and I said, when you first saw this, what did you think? Oh, he said, it's amazing. I said, I rest my case. And so it's always worth talking to the editors of the paper that you're worried about, because sometimes if you say the right thing, you can make them change their minds. I got the Nobel Prize in 1993. I shared it with Phil Sharp. He and I actually both worked at Cold Spring Harbor together for a while. We have one paper mapping, using restriction enzymes to map adenovirus RNA. He went on after and went to MIT and independently did all of this. He had won many prizes for this. For me, the Nobel Prize was the first prize I ever won, and that was because I was never nominated for anything. Jim Watson didn't like me. We had a hard time together, and he never nominated me. Even when eventually he was asked to provide some stuff, He still didn't produce my name and say I was the person who should get it. But, you know, if you've got to get a prize, it's not a bad one to get first. So one of the things that happened with the restriction enzymes was that it was obvious that there was a need for these. The biological community, they were looking for them. People used to come to Cold Spring Harbor, they'd have DNA in their pocket, and they'd sneak out of the meeting, come down to my lab and say, hey, do you have a restriction enzyme that will cut this? Because at the time, we'd found 30 or 40 of these things. And it was obvious that there was a commercial opportunity here. And so I talked to Jim Watson. I said, we should start a company, and we'll use the profits from that company to fund Cold Spring Harbor. He said, oh, you're crazy, he said. Can't make money out of that. Plus, it's really dirty to be associated with industry. And he hauled me up in front of the board of trustees to try to get me fired. But fortunately, the guy who was in charge of the board of trustees was the son of the founder of J.P. Morgan, Walter Page. And he came up to me after the talk and he said, you do what you're doing. You're doing exactly the right thing. So I looked around and I ran into this gentleman, Don Combe. He had started a small company in Boston, in Beverly, and he'd been selling various things, but he was really making a living doing pregnancy tests. He'd set up a basement lab using old equipment that he'd bought from Harvard that they were throwing out. And so I went up, met him. When I went up, this was the entrance to the lab. It was a basement underneath a hairdressing salon. There were 3 people. There was Don, his wife, and 1 technician. And I went up and I introduced the restriction enzymes to him. And he said, yeah, we'll try and make these. And in the first year, he made $200,000 selling these enzymes. $200,000 in 1975. And that was the start of New England Biolabs. We're now a company of 650 people. And Don had the same idea that I had. He wanted to use the profits to support research. And so of these 650 individuals, a little over 100 are now just doing pure basic research. Everybody has to publish. We could be a university lab. It's really a remarkable place. I would also say he did something very insightful when he first put up this building, this one here. First thing he did was to put a daycare center in so that the ladies who worked for us had somewhere to put their kids during the day. They could go see them. It was right next to it. And we did the same thing here in the new building that we moved into in 2005. And we've always tried to be very friendly towards families and, you know, taking into account we give very good maternity leave, we give good paternity leave, and so on. This was the very first catalog which I produced, and basically it was amazing. It had a list of all the known restriction enzymes in it, and then the ones that we were offering for sale, and it formed the basis of the catalog that we use today. I've always loved puzzles, games. I also have a sense of fun. I love humor. I love jokes. I like laughing as much as possible. This was me as the stud muffin of science in 1997 for this catalog. I was Dr. December. I spent my Nobel Prize money putting a croquet lawn in front of my house because I love croquet. Croquet's a great game. It's really one of these games where it's a cross between having some skill as to where you're going to put the board, so it's sort of like playing snooker, but then you need the strategy of chess, and it's a wonderful game. I learned to play it when I was in Sidney Brenner's lab on sabbatical for a year. I've also had a bunch of honors of one sort and another. I got several doctor's degrees and so on. But the thing that really I've done more than anything else is to start a series of campaigns. What's happening to this clock here? Anyway, okay. It's very strange. I mean, yeah, okay. I was rushing because I thought I was out of time, but maybe I'm not. Anyway, I've started a number of campaigns among Nobel laureates because I discovered something very interesting. I've always been something of an activist. You know, I tell people what I think, and if I see things going wrong, I try to tell them that, you know, this is wrong and you shouldn't do it. And people, you know, occasionally would listen, but I never really had anybody do any actions or anything. As soon as I won the Nobel Prize, not only were people listening, they were acting, they were trying to do things. And so I've run something like, probably 45 Nobel campaigns to try to do things. And one of the most successful was we organized the Nobel laureates to get some nurses out of jail in Libya. They'd been accused of spreading HIV in the children's hospital in Benghazi. And it was clear that this was not a deliberate thing. It was bad medical practice because they shared needles. Nevertheless, they had a trial. Judge said, yeah, you're guilty. Several of the nurses under torture said, yes, we did it deliberately. And they ended up on death row. And when I found out about this, I started talking to some of my Nobel friends, and I said, can we do something about this? We got more than 100 signatures. I went down to the Libyan embassy, to the United Nations, because there was no diplomatic relations with the US, or no formal ones anyway. And I talked to the Libyan ambassador. He said, yes, I'll take this letter to Gaddafi. 2 weeks later, I got a call from Gaddafi's son, who said, could you come to Tripoli and talk to us about this? Get on a plane, I go over there. I spent an hour talking to him late one night, and at the end of that conversation, he said, we're going to let them go. And so, we saved a number of lives here, so that was good. And there've been other things that we've done too. This was just getting knighted. They have this sword that they touch you with, and Charles' hands was not quite as stable as you might think. So I was a little bit concerned about that, but turned out to be okay. I also had the pleasure of meeting Queen Elizabeth I've actually met her 3 times, but there was one very memorable thing. There was a thing going on at Buckingham Palace, and she asked to meet the Nobel laureates who were at it. There were 3 or 4 of us there. And we go, and we start talking to her. And it was clear she'd had some preparation, but she was asking really good questions. Very, very smart lady. And then during one of the intercessions, Prince Philip, who was there too, said something absolutely asinine, really stupid. And the Queen looked at him and said, oh, shut up, Philip. She was a good lady. So the campaign that I'm most keen on at the moment is GMOs. GMOs are incredibly important for the developing world. If they don't have access to this technology, they're not going to be able to feed themselves. And this is a crime, and it's a crime because the Europeans who don't need GMOs are blocking it. Greenpeace decided this was going to be something good for them to do. It was the best fundraiser they've ever had, but now they refuse to back down. The leaders won't meet with me. We have 167 Nobel laureates who've all written a letter. We sent it to every embassy down at the United Nations, and the Europeans still won't do it. So you should look at this site, supportprecisionagriculture.org, and if you agree that we need GMOs or come and talk to me about it, sign on. Everybody can sign on and join us. I want to talk just briefly about the importance of luck. So I found out playing billiards, playing snooker, how important luck was. I met Kazu Kurosawa. What a lucky break that was. I got put into a lab. He happened to be there. Best mentor I ever had. Got to Harvard instead of Wisconsin. Looking for eukaryotic promoters in adenovirus. Here we are. This wonderful experiment, and it didn't work. It just failed completely, but we followed through. And so whenever experiments fail, you always should do a post-mortem. You should ask, is nature really trying to tell me something here? Is the thing that we think is correct, is it not correct? That's how you really do well in science, is when you have a failure, find out why, follow through, and if you're lucky, nature's telling you something. Taking an early plane, you probably all know about 9/11. 9/11 was when the first terrorist plane went into the World Trade Tower. I was booked on that plane until 2 weeks before it took off. And I was going to a meeting in California, and the meeting got moved forward one day. And so I had to change my reservation. I ended up flying on that plane one day before it took off. And, if that's not luck, I don't know what is. That is just fantastic. And studying bacterial RM systems, it really made my whole career And I'm still working on them. I still work on DNA methylases. That's what I do. Bacterial DNA methylases is mostly what I do at the moment. And it's been great. I do bioinformatics. It's wonderful. And I just want to leave you with this. I mentioned when we were talking the other day about these books that I think everybody should read. I don't care whether you're a lady, whether you're a man. This first book, The Exceptions, is about Nancy Hopkins and the fight for women in science at MIT. And I think it gives you a very good indication, if you're a woman, of how difficult life can be in the academic setting. But if you're a man, it teaches you, well, you often do things and you don't realize the effect that they have. You know, men and women are different. Women have children. Women have periods. Life is different for women. And I think a lot of us men don't appreciate that and don't take that into account when we're dealing with them. The other book is one that I've almost finished. I'm right on the last. It's called Invisible Women. And it's full of data about how men and women really have very different outcomes, because a lot of science, a lot of experiments, a lot of data looks at it from a man's point of view and doesn't take advantage, doesn't really look at it from a woman's point of view. And the other one I really would like is Breaking Through. Kadi Kariko won the Nobel Prize this year. She's the lady who developed messenger RNA. Her life story is wonderful. It's unbelievable that she managed to survive. It's a great, great story, and she is a lovely person. And this is not a very good picture. I put it together rather quickly. This is a documentary. It's called Picture a Scientist. It too is about Nancy Hopkins, but it's about some other scientists as well. And it is a very, very good documentary explaining the difficulties that women face in science. And I think it's really up to all of us to make sure that we learn how to deal properly with women, whether it's in science, in the workplace, wherever. Because I know after I've been reading this and I look back, I know there are things that I've done that if I'd known more at the time, I would not have done them. And so it's very good. We all need to be aware of this, and women are great. Love them. Okay, thank you very much.
Thank you so much for listening.
Okay.
I believe all of you have learned a lot from Prof. Robert's talk. So now is a more interactive session. So any questions are welcome. Yeah, please use the mic there.
Hi, thank you very much for your talk. You mentioned this conflict with Jim Watson and this relationship. I wonder if you could tell us more how you dealt with it and if you have any advice with that sort of situation.
Well, so I was very fortunate that very early on, I ran into a man called Norton Zinder, who was a professor at Rockefeller and probably was Jim Watson's only friend, and his only real friend, let's put it that way. And I ran into Norton on a I was on a train between Brussels Airport and Ghent going to a meeting. It was the first restriction enzyme meeting. And he was very interested in restriction enzymes, and we became really good friends. And whenever Jim would threaten to fire me, which he did on a pretty regular basis, I would call Norton, and Norton would call him up, and he'd say, you know, Richard's okay, keep him on. And so he was able to calm him down, and so then he kept me going. Later on, there were times when we would disagree. Most of the time we would disagree. He used to get quite abusive at some point, and I used to laugh at him. I just didn't take him seriously. He would, you know, be yelling and foaming at the mouth, and I'd just laugh and walk out of his office. So that's how I dealt with it.
Thank you.
Yeah. Oh.
Thank you. Hi, I'm Lara from the University of Southampton. And I'm so sorry. Thank you for such a wonderful talk, the activism, the book recommendations, and especially the RNA monarch purification kits. Game changer. Thank you so much. My question is, especially with regards to your early career, do you have the luxury of working on one problem, one project, or were you often sort of split between different projects?
So there's sort of two things related to that. One is, yes, I worked on a single project for the most part until it came to restriction enzymes, but just looking and discovering new ones, that was the same. The other is that I don't like people ever telling me what to do. And so if they tell me what to do, I always think it's boring because they thought of it. I want to do what I want to do, and I've always found that this is the way to do science. It's the way I've done science, and it sort of worked out okay. So I don't like people telling me what to do.
Thank you so much.
So, Minnie, don't listen to your supervisor. Yeah, you please.
Thank you, Professor. I'm Ye from Hiroshima University in Japan. Thanks for telling us your experience, like keep bouncing back from frustration and failure. So my question is, during those downtimes in your early career and study, did the idea of quitting up and settle down for something that is good enough came across your mind? And how did you decide to continue your journey? Thank you.
Okay, so I'm a very positive person. So if things don't work, for me, I always think it's because I didn't plan the experiment properly, I didn't do something properly, nature must be trying to tell me something. And I love it when students come and say, oh, I did this experiment you suggested and it didn't work, and I'm always hopeful they're going to make a big discovery. And so I never really got down. I was always a very up person. I'm still a very up person. I love life. I love doing science. I don't feel I've worked a day in my life. I love it. Science is my hobby, I love it. And so even when things don't go exactly as you want them to go, it's usually, and I say in my case at least, it's usually because I didn't plan it properly. And so I will go back and repeat it and plan it again. When we first got this result with a single oligonucleotide at the end of all the messenger RNAs, my postdoc, Richard Gelinas, I said, go repeat it. He did it 2 or 3 times. I said, let me show you how to do it and I'll get the right result. I got exactly the same result, and after that we knew that nature was trying to tell us something, but it took about a year to figure out what it was. But we got a lot of interesting results along the way, so it's good. Doing science is wonderful. There's nothing better in life, I think. It's great.
Thank you.
Okay.
Totally agree with this. You?
Yeah. Hi, my name is Yong Rui, and I'm a PhD student at University of California, San Diego.
Mm-hmm.
So you mentioned at certain points in your life you had to make decisions. Mm-hmm. And I'm wondering if you ever look back at them and regretted them. Like, for instance, you said that while you got a position at Cold Springs Harbor, you also got a faculty position and offer at Harvard. Yeah. And I think most people, at least I would, choose Harvard for its prestige and its influence. So have you ever regretted these decisions?
Never. Okay, because I think when you make a decision, you really have to make the best of that decision. And if you take a positive attitude, I don't care where you go, you will always end up doing something good. And so I never have viewed that I went the wrong way. Luck has always taken me in the right direction, I feel. So I'm very happy. I got a letter from the Unabomber. Do you guys all know who the Unabomber is? He was this guy. He was an assistant professor at MIT, and he was crazy. And he used to send letters and bombs to people. And when my letter came, he wrote 5 letters. 4 people got letters. 1 got a bomb. Wasn't me. I am the luckiest guy you know. I'm serious. I really am.
Thank you.
Okay.
Thank you.
Hi, good afternoon, Professor. My name is Adolphus Lai. I previously was based in the University of Liverpool, and I'm currently based within National University of Singapore as a research fellow. And thank you so much, first of all, for your very positive story. I really enjoyed hearing your life story and where it's taking you today. And my main question is that, okay, so after you got the Nobel Prize, what are the changes, that you feel that has happened in your personal life, other than now being able to do more activist work? Like, what else do you feel has, kind of like, how did the Nobel Prize impacted your life? In one way?
So one thing that happens, as soon as you win a Nobel Prize, everybody wants you. They say, oh, will you come give a lecture here? Will you give a lecture there? I could spend, it's 30 years since I won this prize. I could spend every day of my life somewhere other than home. And so one of the first things you do is you accept a lot of these invitations, and you have to decide which are the ones that you might want to pursue later on. And it takes a while to learn which are the good ones, things that it's worth doing and things that it's not worth doing. But the opportunities are just enormous, absolutely enormous, you know. And so for me, that's been very nice. And I'm fortunate, you know, because I work at New England Biolabs and I help get them set up. And you know, they're very happy with me going around and talking about them and doing things. So they let me do pretty much whatever I want. And because they support research, I'm really a postdoc. To be honest, I'm a postdoc. I do research all the time. I got my computer with me. I open it up first thing in the morning and look and see if some programs have run, you know, and I make a discovery every day. Just tiny little things, but I see things no one's ever seen before. And that makes you feel really good. So that's very nice. I wanted a PacBio sequencer. I wrote a purchase order. 2 weeks later, it came up. I didn't have to ask anybody, did I want it? Because the company supports the research that I do. They're great. They're a wonderful company, really good. Or buy your reagents from us.
Thank you so much, Professor.
You're welcome.
I think you first and then followed by you. Yeah, you first.
Hi, I'm Alina from ETH Zurich in Switzerland. Thank you so much for your talk and also for sharing some of the activism that you used your position for GMOs and the Libya situation. Would you mind sharing some of the other things that you've done?
Well, let me preface this by saying that we don't always have immediate actions ourselves, but rather because there are a lot of Nobel laureates supporting something, very often people who can do something will be motivated to do it. And so there's been quite a lot of times when that has happened. So for instance, in the Libyan situation, there were only 2 countries that had good diplomatic relations with Libya. One was Britain and the other was Egypt. And I talked to the Foreign Office in London, and they were very helpful. They arranged for me to stay at the British Embassy when I went over to Tripoli. And they did a lot of work behind the scenes. They got many European governments to support what we were trying to do. And so I think this is the real value, is that if you've got, you know, 10, 20, 30 Nobel laureates all agreeing to do something, people listen. People go off, and they often do it. But not always. So we tried very hard to get Aung San Suu Kyi out of prison in Myanmar and out of house arrest in Myanmar. We had 230 Nobel laureates signed a petition to try to get her out. I got that delivered to the generals in Myanmar, and it was without effect, total no effect whatsoever. One of the first things we did was to push for open access publishing. And we got a lot of laureates doing that. It's taken a long time, but it's coming. We're almost there now. And we started by trying to persuade Science and Nature that they should make their journals open access. They should charge for the news and views, but they should make all the papers open access. And they refused to do it. If they'd done it, I think open access would have been the norm 20 years ago. But it wasn't. But it is getting there. We are getting there. And I think it's outrageous that the publishers should make money from taxpayer money doing the research that's, you know, you want everybody to know about this. You don't want to have it behind a paywall. I think it's disgusting.
Thank you.
Okay.
Hi, I'm Kanishka from National University of Singapore. Thanks for sharing your lovely insights, Prof, and also the great work that you're doing. So going back to your pathway, right? You said you started under a hair salon, basically, right? Do you think this is possible in the modern day example for us if we want to, having faced many obstacles and if we want to come up with our own solution? Why I'm asking this is, one is like example, if you want to get something published, it's getting a lot more challenging because, for example, Nature, they want single-cell RNA-seq data or now even spatial and things like that, which cost a lot of money. And for early careers, it may be challenging to have access to those type of funds and even grants. So just wants to share your insights, whether it's more challenging now compared to your days.
Yeah, but again, why do you want to publish in Nature? Why do you want to publish in Science? These are the journals that have more fraud than any other journal I know. Right? Why do you want to publish? I will not put my name on a paper going into Nature. I have 2 papers in Nature journals, And in both cases, I wrote in the acknowledgement that I would normally have been an author, but I don't agree with their open access policy, so I won't do it. And you don't need to publish in these journals. There are plenty of really good journals where you can publish. And it's the quality of the paper that matters. It's the quality of the work you do. There's the bioarchive. You can always put your material in the bioarchive. If you're a physicist, there's the XArchive. There are alternative ways in which you can make your work known. And when you face, if you send in applications, face committees, you can always put a letter saying, you will not publish in Nature and Science because you don't agree with their policies on open access. And you can say, hey, but I've got 6 papers here that are really good. You read these and you'll see the sort of fellow I am. And MIT, I might add, are now not looking at the journals you've been published in. They're not looking at open access and so on. And the guy who came up with that, I actually wrote an obituary for him because I think he did more damage to science than anyone I know. And I actually told him that to his face too. He was terrible. It was Eugene Garfield, his name is. He was a terrible guy, just terrible. He's the reason why all of these universities and so on look at impact factors, because they like numbers. All these wretched financial bureaucrats are terrible. They just want to see numbers. They don't want to actually read and find out whether you're any good or not.
Thanks.
Okay.
It's very good to know. Thank you.
Hi, Professor.
Hi.
I am from Malaysia, from University Tunku Abdul Rahman, and I have something to ask you about, just how you mentioned about luck. So we know that everyone will not be so smooth in their life or career path. So any advice, or may you have any share or any comment for those who are in not so lucky?
Well, I think everybody is lucky. And, if I would challenge all of you in this audience, go back, look through your life and see where luck took you one way rather than the other. And you can say when a lucky break occurred, did you take advantage of it? And this is a lot of people don't. A lot of people feel guilty. They say, oh, well, I was lucky, but this person was not. And when you're lucky, you've got to take advantage. Everybody has luck. So, I don't believe that people are unlucky, but some people have more luck than others. I happen to be one person who had a great deal of luck. I've been really lucky. I'm not smarter than everybody else. I'm not a great genius. I'm not an Einstein or anything like that, but I've been really lucky. And I've taken advantage of it, and all because this old man spoke to me when I missed a shot at snooker. It made a difference, really made a difference in my life.
So I'm lucky to sitting here as a moderator. So actually I have a question. So sitting here as a female scientist, supervising students for I think 12 years.
Mm-hmm.
So especially in recent years, I always facing a question from some very talented female scientists, students. They were saying that they see no hope for being a postdoc in the near future because the money they earn are not even sufficient to hire a maid to take care of their children. Right. So that's why they somehow giving up half the way. So that's why if you look at the number, the leaking pipe is quite obvious in Singapore local ecosystem. So sometimes they cry in my office. So I try to use like 1 or 2 like sentence to kind of explain. I don't think you should give up, but always feel like I couldn't find the right words to express. Do you have any?
I'm not sure I have the right words, but I think, when you're mistreated, and for an awful lot of women, they're simply mistreated, complain. Go and talk to. This is what Nancy Hopkins did, right? She goes to MIT, and they give her a lab half the size of a man. They won't give her a secretary, and half the men thought she was a secretary when she first arrived. It was tragic, the way she was mistreated. And she's a lovely person. She and I are very good friends, and she throws wonderful parties, I might say. In my lab, we used to, every time a postdoc would come and run a gel for the first time, they had to give a party. And when Nancy came and worked in my lab, she threw the best party. There was champagne, there was strawberries, it was fantastic. But she was incredibly mistreated, but she did something about it. She went and talked to the president of MIT. She gathered other female professors at MIT, and they started measuring the amount of lab space they have and the resources that they have. And the president agreed that they were being mistreated, and he did something about it. So I think the answer is that when women come and tell you, you know, it really is not right what's happening, I don't get enough, I can't do it, find the right person for them to go and talk to and get things changed, make changes. I should say there's an award called the Blavatnik Award in Israel, and you have to be under 40 to qualify. And I discovered I had worked with a student who had been at Harvard and then went back to Haifa, and I nominated her for this prize. And the committee wrote to her and said she was 40 at the time, she had 3 children. And they wrote to her and said, well, you're too old, right? And so, you know, I talked to her a little bit, and in Israel, you take time off when you have a child, right? And so a man who is 40 has had more experience than a lady who is 40 who has children. And the Blavatnik people changed the rules so that they would take account of the fact if women had had children, they could be over the 40-year age limit. I think there are times we can do more and more about this. You know, this meeting here, when I first looked at the program of speakers, there were 14 men and 1 woman. You know, this is no way to provide role models for you students, right? If you're a female, you want to hear about good women, women who've done really well, so that you can learn something from them and see how they've overcome these difficulties with children or whatever else goes on. So I think I've been talking to the head of NRF about this. So.
Thank you. Thank you so much. Sorry. Oh, I guess. Okay, please.
So I am Adira from Shivanadar University, India. And thank you for sharing your wonderful life journey, sir. Actually, I wanted to ask, you have said that you are a positive man and you are always be positive. So can a human can always be positive in some point or some in situations for family or for friends? We have to do something. We are forced to do something else that we don't want to do. So in such kind of situations happened in your life? Or
I've not really had problems like that. I think the worst thing that happened was when I failed physics at A level, and I had to spend another year at school. But I didn't really worry about it. I just decided, you know, I knew why I'd failed, and so I got over and made sure that I did it. And that 3rd year, because I'd already got 2 A levels and it was easy enough, it didn't take a lot of extra. So I used the time. I did more chemistry. I had fun. I did a lot of reading, went caving a great deal. I had a lot of fun. You know, if I run into something that looks as though it may be an obstacle, I find a way around it. And I'm looking positively to find a way around it, because maybe that obstacle is going to lead you into a new direction which is going to be more productive than you would have been otherwise, right? This is how you stay positive, right?
so what do you think, why all of them can be positive every time? Some are.
I'm positive every day, I'm sorry.
So what is the reason behind it? Is there anything you can suggest to others, those who are thinking negative sometimes when they are failing?
Yeah, well, but I really think you have to remember, failure is a good thing, right? You know, Asian parents often tell their kids, oh, you fail this, you fail that, you're terrible. It's not true. Failure is when you learn. If you never fail, you never learn. You know, Bill Gates, the first company he tried to form was a failure. He did okay, yeah. You know, failure is a good thing. I talked to some students in Dubai, some young kids, not too long ago, and I was talking about failure. And one of the kids contacted me. He sent me an email, or his mother sent me an email, and said he wanted to talk to me because what I'd said about failure had changed his life. And it turned out he was very good at rollerblading or something, and he could do all sorts of tricks. But there was one trick he wanted to do, and he was afraid he would fail if he tried it. And after he'd heard me talk, he said, I decided I'm going to try it. And he tried it, and it worked. He didn't fail. And he was overjoyed. Never had a better conversation with a young man than I had with him. And it was all because, you know, he got the message. Failure is okay. There's nothing wrong. What you don't want to do, though, is keep failing by making the same mistake over and over again. You work out why you failed. You know why it's so safe to fly in airplanes? Because every time there's a crash, they do such a deep post-mortem to find out why, and then they fix everything. And that makes flying one of the safest things you can do. It's much safer than walking across the road, for instance. Many more people are killed going across the road. Doctors, on the other hand, tend not to be that way. Doctors are told and taught in medical school, you don't ever want to admit that you failed, because then you will lose the trust of the patient and you will not be as good a doctor. I think it's nonsense myself. I think if you fail, you admit it. You say, I screwed up, you know? And I think honesty is much, much better than trying to cover up failure. Honesty is a good thing.
Thank you so much, sir.
Okay.
Do you think some students maybe need a more caring supervisor? So that's why
Absolutely.
they get somebody to talk, so you are not alone.
Yeah. But I think, you know, one of the things, if you're a scientist, you need to talk all the time anyway. Okay, science is not a very solitary activity. It used to be, but it certainly isn't now. And the nice thing about science is that wherever you go, if you come across another scientist, you've got a friend. Okay? And we can disagree about all sorts of things, and we can argue about hypotheses and other things, but we never need to shoot one another in order to prove that we were correct. And I think it would be better if the politicians took the same sort of attitude.
Please join me to thank Prof. Roberts again for the very wonderful talk. And
Okay.
yeah, thank you so much.
Thank you.